Institute of Malaria Research. Please, Dr. Rajender, please join. So, at the outset, because of the paucity of time and some of our colleagues have to leave, can we switch off this one? IT one is there. Okay, so at the outset, I request Dr. Mohan G.H, because he have also to leave quickly. Dr. Mohan G.H is doing a lot of activity in the National Center for Biological Science. I request Mohan to please come and explain in 5 minutes about your work and about the laboratory animal welfare program.
Hello everyone. Thank you Dr. Singh for inviting me to share thoughts about lab animal welfare. Myself, I'm Dr. Mohan. I'm in charge of Animal Care and Research Resources Facility at NCBS, the National Center for Biological Sciences. We are actually, at NCBS, we call it as ISAE, Animal Care and Research Resource. We have over 400 strains of mouse repository, and then over 22 lines of rats, and we have zebrafish. And then our main motto there is to provide specific pathogen-free animals to ongoing research. And not only that, we train the users. First of all, educating them is very important. So, main mantra is to have more training, less assisting. So, anybody who is coming into animal research, we see them, first they are trained about animal welfare, the guidelines, especially our lab animal science guideline, like the 3 R's concept, Rosenberg's 3 R's concept, that is replacement, reduction and refining. Of course, now CPCSA has also introduced about reuse or rehabilitation. And then, about this is about animal ethics. And the second, the objective of the facility are to educate the users is about how to make that the 4th R. For our purpose, we call reproducibility as a 4th R. Like, how to achieve reproducibility in your animal research. So, because biological research has a lot of variability, because these animals, especially their health status, their genomic listing, is not constant. That is why we keep them in controlled environment, and we should monitor their health status and genetic quality. So, and not only that, environmental variations will definitely affect their phenotypic, and indirectly or directly it will affect your experimental outcome. So, one way is to educate the users as well as the facility staff and whoever is responsible in care and use of these animals. So, we conduct workshops, and for the technicians, we conduct workshop for the beginners who are into using lab animals in their research. So we concentrate more on humane handling, restraining, biomethodologies like different injecting, the animal dosing techniques, blood collection techniques, necropsy and euthanasia, anesthesia, all those things. So we conduct that at least 2 workshops every year. So this is coming on the website and also on the WhatsApp platform. So anybody who is interested can apply. So this is what we are doing at NCBS.
Sir, can you elaborate on cryopreservation?
Yeah, that is another section of our facility. It's called mouse genome engineering facility, where we do generate our genetically engineered mouse models using CRISPR-Cas9 technology, and we do cryopreservation, sperm cryopreservation, embryo cryopreservation. We do rederivation, especially if you have a strain which is non-SPF origin. To bring it to SPF quality, we do rederivation technology, in vitro fertilization. All these services are also available for external users, so you can avail yourself.
And this regular workshop that you are doing for genetic
Exactly, exactly. Same thing. Whatever these services, we do conduct, the training, customized trainings, and also in the form of workshops. So you can see our website. You will get lots of information about these services. Please feel free to contact us. We are here to give whatever the support, the research resources in terms of animal resource as well as other training resources from NCBS.
And about marks,
yeah. So this is the recent one. So these days, we were doing about animal users training, and also for the technician, those who are into beginning of using animals. Now we felt like, to also, because in the veterinary, this thing, exposure to lab animal science is little, it's minimal. So we have started a course, kind of a basic training course, like 5 days course for veterinarians who, it's kind of giving a practical exposure to lab animal science for the veterinary graduates, so that, we concentrate more on giving the managemental aspect of lab animal facility. Also, there will be interactions with users, because first of all, you should understand what is the expectations from the users, and then of course we will also give you about the managemental practices, and there will be high barrier facility visits. So this is all the plan. So we can make it.
Do you? I hope if you have any question, please feel free to ask, because there are a lot of new veterinarians, and I am trying to expose you to the best places and possibility. And NCBS is one of the best places where they are doing this high-end genetic manipulation of the mouse, micro injection, cryopreservation, as well as the training program. Because as a vet, we never had any opportunity where we can directly go and hold this training program for us to get competent into laboratory animal science, because it's very highly scientific. So, sir, this March program is still open for veterinarian, or is it only for?
It's still open. It's still open. You can apply. Yeah.
So, is it only for veterinarians or even pharma graduates?
Yeah, this one is purely we are concentrating for master degree holders, those who have finished masters in veterinary science, and since we have started as a first course, we are limiting that seats to 20 only. So concentrating more on veterinary graduates. Postgraduate.
But you hold program for non-vet as well.
Yeah, so like non-vet pharmacology.
Yeah, that is the second one, that which will come in June. Sometime in June or July we are going to advertise. That is a routine thing for the users, the beginners who are into lab animal science usage as a user, and also for technicians, those who want to get trained in biomethodologies. So those.
So basically,
yeah.
During the that course for veterinarian, and since when that
ask slowly and asking speaker. No
Yeah.
Problem.
Yeah, the course duration, what we have fixed is like 5 days, week course. Usually it will have, we will try to cover some, the important theoretical aspect in the management, and to understand animal biology and kind of. We concentrate more on practical exposure, like, training them for humane handling, biomethodology, especially interacting with the users, the lab requirements, and also exposing them about the managemental aspect of high barrier facility, how in the latest modern, animal house technique, maintenance, all these things, especially the managemental aspects we are concentrating.
Is your answer clear?
Yes, sir.
Because that veterinary means it's only as a veterinary curriculum, we concentrate more on treating domesticated animals or wild animals or pet animals. But lab animal science is entirely kind of like remote for us. So we try to give that kind of exposure, what is expected of the lab animal veterinarian. So lab animal veterinarians are like kind of, you have to do all-round work, not only as a vet just to treat the animals, also as a manager. And it's your role as a full-time lab animal veterinarian to interact with the administrator and also about the ethical aspects and to ensure that the proper animal usage happens. I mean, like educating more. It kind of like it's a teamwork. So that kind of exposure will be given to these guys. Freshers, so that if they wish, they can take up as a lab animal veterinarian course, or if they take up as a research. Then veterinarians are better placed to take up animal research, especially they do wonders because they know the physiology, the biology of animals very well, and yeah, that is how it's a kind of a practical exposure to them.
Any other question? Yes, sir.
So, which pathogen do you screen for in your health monitoring program, and what is the duration?
Yeah. So, for health monitoring, we are actually right now in-house. We have not started yet. It is outsourced, but we are following the PHILASA guidelines. So, for level, we have 2 levels of health monitoring. Usually, PHILASA will ask for the quarterly health monitoring. So, and it depends, your exclusion list depends on 3 criteria. Number 1, zoonotic important pathogens. The second criteria comes the pathogens which have got high virulence, the mortality rate is so high. When the infection comes, especially in lab animal science, if very less scope for treatment. Because once the infection comes, by the time you treat the animals and they recover, they become useless. Or this treatment, your own treatment may cause as a variability. I mean, like it's an unwanted variable in the experimental testing. So, and then the third group is decided by the end user. Like, those pathogens which will not affect the animal health nor the handlers or the animal staff there, the users, but it can affect your experimental outcome. So based on these things, we decide our exclusion list. It is a relative term, for our facility, we follow a global list. About 46 pathogens we screen every quarterly. Very expensive thing. But each facility has its own criteria. Especially the last list of exclusion list pathogens, what you include, that will decide the whole. Because primarily you should first screen for zoonotic important pathogens, and then the second one is highly virulent ones. So then the third group, based on that, you will exclusion list will decide. It is PCR based. It's actually multiplexing, but if there is any positives found, then it will be PCR based to confirm it.
Any other query? Yes, Green.
So you can feel free to write to us. You can visit our website. It's not displayed. If you visit NCBS website, you will see ACRC. So all the information is there.
No, sir. NCBS in Bangalore.
Yeah.
Okay, sir is in Bangalore. It is nice weather, beautiful campus, and he's a great host. And I saw also every facility there is very high end. So when we were handling, it was in the containment IVC as well as the cage changing station. So that's why I think yeah, very, very nice place for the vet to get exposure for lab animal science. So sir, thank you very much. You are getting late.
Yeah.
And very fortunate you holding for so long time.
Thank you. And then, Professor Vijay will be there with us for the next workshop also, because we have experienced this, especially when biostatistics is coming. So even I was talking to our secretary, Datta sir, for all the IAEC nominees, we should have some biostatistics course, because always we try to minimize the number rather than have some proper justification to ask for the number like that. Anyway, things will take time to improvise.
I request Aina and Priyanka to facilitate sir with the bouquet and the note, because he will be leaving. Portraits. I popularly call him Kannadiga hero. He is so stout and strong and all like that. Thank you very much. Okay, sir. Nice to meet you. Now next in line is our Owen, Dr. Rajender Motiani.
Dr. Rajender Motiani was previously in IGAV, and some of our younger colleagues from Jammu, I don't know whether they came. Anybody came from Jammu? Or okay, you were coming. So there was a question that can you introduce us to some innovative, energetic, different way? So I asked Dr. Rajender. He's innovative. He's a veterinarian from Nagpur Veterinary College, and thereafter he did his PhD from US and publishing very high impact paper and also very high competitive grant, including I think that's a Welcome Trust we recently got the Welcome Trust also grant, I think, which is topmost grant for the researcher. And I request Dr. Motiani sir to please introduce and also inspire the young veterinarian and also related to theme if you would like to talk.
Either there or the podium there.
Standing is better. So, thank you, Dr. Vijaypal, for such kind words. So, yeah, I have a story. I mean, I don't know how dynamic I am. Can I match the levels of Dr. Vijaypal? only time will tell, right? Because he's still too young. So, anyways, I mean, my journey is different. Yes. So I started. BBSc in Nagpur Veterinary College, and this was early 2000. I started in 2002. And very early on, when we were in still preclinical courses like microbiology and physiology, biochemistry, I realized that I like it more than going to dispensaries. It's not that I didn't went to dispensaries and didn't carry out work related to medicine or surgery, but somehow I realized that I like spending time with microscopes. I like looking into what is going on at a more detailed level. So very early on, I think from third year onward, I started working in the lab. And the way I started was, there are always lot of MBSc students, right? They are doing their research. So I started shadowing some of them, like what sort of work they are doing. And most often their work was, at random times, right? So, if some of you are, doing MBSc or have completed, you would realize that many a times during day, they are taking practicals, right? They are teaching UG students. They will do experimentation towards late evening, sometimes in night. So that actually time was a blessing, because during the day, I was taking UG classes, and during evenings and night, I was working with MBSc scholars. So what it led to, I was exposed to research techniques like PCR, and a lot of, at that time, MAPSU, especially Nagpur Veterinary College, got a couple of good grants from including DBT. So what it led to, we already had a tissue culture facility in our veterinary public health department and a microbiology department. So with that, little bit exposure, and we did some work. Now looking back at it from where we are today, it seems like that was maybe very trivial, but it was very important, because that exposure, early exposure, helped me in understanding what is my actual liking. And I realized that research is the one that I, kinda like more. And then I continued to work in lab. We published some papers as well in international journals from Veterinary College in Nagpur. And then, towards the fourth year, I decided that I have to go for higher studies and research-oriented studies. So then I started working on, GRE and TOEFL, which are kind of entrance exams for graduate programs or PhD programs in US. So eventually, the internship and all ended in July, somewhere mid-July, and in August, mid-August, I took a flight for New York. I did a PhD from Albany Medical College, New York, and that experience and the research that I did in the veterinary school not only gave an exposure but also helped in building a CV that was appreciated by international researchers, right? And allowed me to start my research career over there. So I did my PhD in New York. I stayed there till end of 2012, and for personal reasons I came back. And you always struggle what to do. You have some personal responsibilities. You have to come and cater to them. And there is the other call within you that I want to do good science, right? But if you continue to try good things, things happen. So वो कहते हैं ना शिद्दत से चाहो तो सब हो जाता है eventually. So what happened at that time? There is this call now. It's quite famous. Inspire Faculty Program, which is post PhD. There is a good fellowship that they allow you to work in partially independently. So I got that fellowship, and I was again looking for where to go. And fortunately enough, I ended up being at IGIP. Met at that time the current director, Shavik Methi, and the ex-director, Dr. Rajesh Gokhale. And then I worked with Dr. Gokhale for around 5 years. And since 2018, now 4.5 years, I am leading my independent group at Regional Center for Biotechnology, Faridabad. And the areas of my research interest are looking into cancer biology and skin pigmentation disorders, and majorly ionic homeostasis, how ions, specifically calcium, contributes to these diseases and how we can use them as a target for therapeutics. And I extensively use animal models to understand, and I try to follow as much as possible the 3R rules of animal welfare. And we extensively work on mice and zebrafish, and we have in past, along with Dr. Vijaypal, have also worked on buffalo, wherein we tried to use buffalo as a model system to understand vitiligo, the patches that we have in humans. If you guys would have noticed, some of the buffaloes also have these white patches, not on the hair, but on the skin. And what we found while working with Dr. Vijaypal, that they are excellent model because they actually recapitulate whatever is changes that are happening in human skin. So we know that some of the other colleagues are kind of using it as a model system to identify novel drugs to take care of pigmentary disorders. And with this, I would be happy to take any questions that you guys would have. Thank you.
Any questions? So those young veterinarians who wish to pursue these these studies abroad, sir, because we are never been trained so means in a way that actually required there outside. What should they do if they want to go?
Start early. The first thing would be start early and work hard. Because as I said that you have to utilize your time well. During day, because you can't compromise on your UG grades, right? Because they look at a complete package. When in our system, we look at if someone was able to clear an entrance exam, right? Over there, they look at your complete CV. You write a letter of statement or statement of purpose, SOP. So they want to look what all you have done. And for achieving other things, if you compromise on your UG grades, that won't help. That will reflect bad. Actually, you have to maintain everything. Your UG grade should be good. If you can work in labs, I'm saying that this we were able to do some 20 years back. So I'm sure today the MBSc projects and the kind of work that is going on in several of our veterinary colleges would be a lot better. And the infrastructure is improving. So certainly there are possibilities if they can shadow MBSc scholars, work in the lab, get some experience, and if they are able to contribute, significantly, I'm sure that they might also end up authoring some paper, and all this collectively helps in getting admissions in good international universities and institutes.
Which animal model you are using in your cancer?
So we use mice and zebrafish. So mice typically used, everyone uses it, but we are also using zebrafish because it's a wonderful model. It's a transparent embryos, and we have a transgenic lines wherein the blood vessels are green. okay, and we inject red cancerous cells in them, and we try to follow where they are going. So if we have to do metastasis studies in mice, it becomes very difficult to image where exactly cancer cells are metastasizing. And if you want to identify a particular factor that contributes to cancer metastasis or secondary site movement, zebrafish serves as a very good model system. So we are using zebrafish specifically for that, but for all other xenograft studies and all, we use mice.
Any other question? Okay. If there is no question, he don't remember Dr. Rajender Motiani, but we worked together in a buffalo model to prove the buffalo is the real model for vitiligo. And we both are the vet, usually vet, if they are two together, they fight. It's like male, but we were, I think, very good.
Yeah, it went very well. I mean, I just mentioned to them, it was...
Because he was very good in molecular biology, I was good on the rest of the things. But then our director sir was very, very great scientist and great human being. Now he is the secretary DBT. He wrote a kavita on animal welfare. कि कई की बात कह रहे गाय हमारी माता लेकिन भैंस में कोई पाप थोड़े किया तो ही राइट ए कविता सर तो मैं उसका सुनाना चाहूंगा भैंस की व्यथा भैंस की जबान से
भैंस की व्यथा भैंस की जबान से बच्चा जब बड़ा होता है दूध मेरा पीता है वो भी बोर्नवीटा डाल डाल कर और निबंध लिखता है गाय हाथी और कुत्ते पर क्यों अगर बच्चा लिख नहीं पाता तो बोलते हैं काला अक्षर भैंस बराबर तो दूसरे जानवर क्या पोस्ट ग्रेजुएट हैं यदि कोई गलती करे तो लोग कहते हैं गई भैंस पानी में कहते हैं ना कि गई भैंस पानी मतलब गड़बड़ तो बाकी जानवर क्या कोका कोला में जाते हैं कोई न सुने तो कहते हैं काला भैंस के आगे बीन बजाना कोई ना सुने तो भैंस के आगे बीन बजाना डॉक्टर संजय ने बताया था सुबह सुबह जैसे वाइफ के आगे क्या सुनत बीन बजाने पड़ता कोई ना सुने तो कहते हैं भैंस के आगे बीन बजाना बाकी के आगे क्या लता मंगेशकर का गाना बजाते हैं तो क्या बिगाड़ा है हमने कौन कह रही है भैंस तो हम कई बार क्या किसी स्पीसीज से अट्रैक्टिव हो जाते हैं गाय गाय ठीक है बढ़िया नो प्रॉब्लम लेकिन भैंस भी बेचारी उसी में इतना ही जी है हम तो वेटरनरी हमको समझना पड़ेगा उनको भी इतना ही दर्द होता है तो ये हमारे सर ने लिखी थी आज वो मैंने आने को कहा था बता नहीं पाए और मैंने डॉक्टर राजेंद्र ने उस टाइम बड़ा अच्छा उत्साह से काम किया
If any one of you have any question,
सर एक और मुहावरा होता है ना अकल बढ़ेगा भैंस
हाँ आप इसको बढ़ाओ आगे अब आपका नाम क्या है
सर प्रेरणा
प्रेरणा बंसल आपने ईमेल पे बहुत क्वेश्चन पूछे थे आप ये लो और आगे इसको बढ़ाओ इस कविता को थैंक यू सर
so yeah, I was just saying at the end that if you guys have any questions, please feel free to write me. All the details are available on my official web page, and you guys can also find me in all sorts of social media.
Yes.
Thank you.
Yeah. Use social media for good work. Find him on the Facebook, tag him or something like that. He will respond. Usually respond. Our next speaker is Dr. P.K. Atul, Pravin Kumar Atul. And while I invited him, he is very, very philosophical sound. And one point of time, I was working in Food Safety Standard Authority of India as a joint director. I was feeling very, very low. Suddenly, by mistake or I don't know why, his call came. And he spoke so nice and explained my situation. And his interpretation of the things, especially the animal welfare, are different. So over to you, sir.
So thank you, Dr. Vijay Palsi. नमस्कार फ्रेंड्स अभी हम भैंस पर बात कर रहे थे उसका सम्मान करना भी चाहिए देश में महिषी देवी या वन महेश की कल्पना से सभी लोग बहुत लिखता भी है आर्कियोलॉजिकल साइट पे जाने से दिख जाएगा कि मैं इसमें अपनी बहुत अर्ली लाइफ कुछ जानता नहीं था सिर्फ पढ़ाई की थी और उससे तो 1-2 साल मैंने वेटरनरी साइंस में वेटरनरी हॉस्पिटल की सर्विस की थी तो मैं अपनी प्रोफेशनल लाइफ और उस समय तक शादी वादी भी नहीं हुई थी तो कुछ भी वास्तव में नहीं पता था। तो यहाँ दिल्ली आया था, दिल्ली स्मार्ट लोगों की जगह, वेटनेरियन को यही समझा जाता है कि बस क्या यहाँ एक बड़े स्मार्ट दोस्त, अच्छी दोस्त बनी बात में, बहुत ही लर्नेड पर्सनैलिटी बनी। उन्होंने देखा कि वेटनेरियन है, ज़रा देख रहे हैं कि क्या रख लेगा। तो उन्होंने कहा हेलो डॉक्टर, तो खावा दी, तो शेक हैंड किया। तो दिल्ली में अगर ऐसा नहीं करता हूँ तो बैकवर्डनेस हो जाएगी। तो मैंने शेक हैंड तो कर लिया। उन्होंने कहा आपको कैसा लग रहा है, कैसा लग रहा है डॉक्टर? तो क्या बताएं कैसा लगा मुझे, तो अभी तक यही पता है भैंस की जुबान पकड़ रखी। तो भाई भैंस की जुबान पकड़ देखते थे आपका हाथ भैंस की जुबान जैसा लग रहा है। तो ये तो भैंस के विषय में सम्मान में बात है, तो आपने कहा इसलिए उधर दिमाग से ला गया। तो इस तरह से सर्विस यहाँ दिल्ली में शुरू हुई और मैं currently I am working in National Institute of Malaria Research in this field. Right now I am developing a model facility. Instead of facility, I named it test research laboratory, in vivo animal experiment laboratory. So it is my humble appeal to each and every professional whoever involved in the lab animal science and working in the houses. So it should be renamed. This animal house should be replaced. No research is to be possible to conduct in house or animal house. It should be called as a laboratory, since it is a full-fledged and established science. The animal science or veterinary science. It should not be called as an animal house. It's a challenging task, but easy. And hopefully India will give this term to entire globe and this scientific community as an in vivo animal experiment or animal test research laboratory, or we can more appropriately, it will be an in vivo animal experiment and animal welfare laboratory. It should be named that. So since Dr. Vijay Pal is LASA president, he has to raise this wire, CPCSTA, to change this in the honor of lab animal science for animals also, right? So in malaria research, what I am doing right now, developing this model facility, hopefully it will start this year. And you may visit at any time in this building. It's a compact building and it's different. So in that building you will get a different experience. So Dr. Matin is here, we have seen it. So in this malaria, previously for 15 years I was dealing with this lab animal science, animal welfare like that. Right now I am dealing with the malaria parasite biology, especially animal malaria parasite. And these parasites start from the amphibians, infection found in salamanders, after reptiles, the snakes, Plasmodium promodoni, and afterwards the avian species, lizards, and avian species, around 600 avian species are found infected with the Plasmodium parasite. And afterwards the smaller mammals, rodents, and rodent parasites are. Only three things are required. So by acquiring the knowledge, if you are interested in malaria research, you may go to, if you think only about animal model, you will not go up to the sophistication level. You have to go for the animal, which animal will be suitable for this particular work, which kind of vector will be suitable for this work, and which parasite. So this is, and currently I am doing about the primary screening of compounds for the anti-malarial activity by choosing rodents, laboratory mouse, most widely used model, even laboratory rat also, and depending on some toxicological work in guinea pig also. And for vectors, for mammals, anopheline species, Anopheles mosquito, for avians, Ulyssine mosquito, Ulex and Aedes mosquito, and for other non-human parasites, simian malaria, the locust monkey, cynomolgus monkey, and rhesus monkey are in use. So there are very limitations regarding malaria research using the non-human parameters. So meeting out, we can go up to the perfection level and up to the desired results and expected outcome. So with these things regarding animal welfare, I think how simple है, एक combined approach है. Animal welfare is a combined approach. इसमें not only kindness and compassion, but in it includes 4 E's, E alphabet, 4 E होते हैं. पहला environment, consideration of environment, for the comfort of animal. Second, the ecology. You must know about ecology, just to ensure the comfort of the animal. Then ethics, whatever our rule regulations, obligations, and our moral obligations, ethical considerations, and then finally economics. Having these 4 E's, along with kindness and compassion, we will be able to ensure this animal. If anything less, we will not be able to ensure this animal. This is all about. If anyone is interested to come in the malaria research, you can come, you can join the PhD program, even if you come for massive dissertation work, if you are interested in malaria. I will be there to help you in all this. Thank you. Thank you.
So, can you start the presentation, Dr. K.V. Patel?
So, until the presentation is loaded, I will get started. So we started good morning and close to good evening, correct? How many of you are veterinarians? Oh, very many. So I am also a veterinarian. I completed my graduations in 1995. Then thought, okay, I will not be able to do so much clinical because of my body structure and physical little issues I faced during surgery. And then decided to pursue animal nutrition. So I did my animal nutrition, and then, as usual, veterinarian after doing it is without not finding a job. Then what to do? So started with Society for Prevention of Cruelty to Animals, Baroda, and there I have been introduced with animal welfare. Could not stick to that place for long because bread and butter, tummy was not getting full there. So I moved in lab animal science, did some practice, and since then, you can say, since 2000, I am in lab animal science. So, my presentation. So first of all, thanks to Vijay for inviting me, and my objective to come over here to learn whatever I forgot in veterinary science is a large animal. When I came into lab animal science, like we learned small animals, rat, mice, hamster, guinea pig, rabbits, then moved little ahead to back to veterinary means dogs. Little experience learning with NHP, non-human primates. So this is what the umbrella, but forgot cattle. Okay, and then I realized, okay, this is the opportunity to go back to the vet school, and Vijay has organized this, so I felt really happy to come over here, and then NDRI team, Dr. Kamboj and all, they justified it really. Here should not touch to the top of the roof of the cage. So height is whatever mentioned in Indian standard is little lesser. When your animal rabbit grows more than 3.5 kg, we will touch it. So these are the behavior parameters. So second point is this space requirement, enclosure requirement. Now I would like to bring the third point, enrichment. What is enrichment? In a natural environment where animals are living, they have some behavior pattern. To meet that behavior pattern, when you enclose them for a research purpose, you don't provide them. For example, rat has a habit to hide, go into burrow. Okay, so they need to provide this kind of natural environment. Then only the physiology will be normal and your research data would be normal. So this is written in an international regulation, enrichment for all to meet species-specific behavior. This is required. When single-housed animals, when they are social animals, we are required to house for some reason, for breeding, for experimentation, individually, then they need to provide some aid, let's say toys, tunnel, to have that species-specific behavior explored. So second point is this, and ELA is this guide and other international standards say not only for the individually housed animal. This I learned very recently. Even group housed animal also should be provided. Okay? That if there is a hierarchy, fighting happens, then these toys and other things, whatever happens in the natural environment, they provide us base for hiding. Then another point, social housing. This has been discussed in NDRI team has discussed very much. When you separate what happened, production, we feel it is improving, it is not improving. Same way, social species should be housed socially as long as it is possible. We generally believe male fights, separate them, don't house them, but not so. You can provide tactical context. There are very many ways to improve the animal welfare. That was my fourth point. Then veterinary care program. So euthanasia, American Veterinary Medical Association Guideline 2020. Okay, they revised the guideline when the new evidences comes in. So people were discussing in the morning, okay, welfare should be science-based. Means science is evolving, data coming up, and welfare standards keep improving. So latest guideline we have to follow. Then pain and distress management, human endpoint, these are also very important. So basically, the veterinary care program is very important to have the best animal welfare. And finally, IAC means institutional level committee, where in Indian scenario, non-scientist is not mandatory. But like in an international scenario, non-scientist is must, who is representing a non-scientific view, a layman view. Okay. Similarly, another point, another member should be non-animal user. He has not used animal. He represents the common public. So these two members, when they are there in the committee, they really have a total public opinion when protocols or research is discussed, and then the solutions comes in, whether the project is approved or not approved. So these are the points I would like to emphasize with this crowd, and I think I have taken more time than expected, but I thought that this is required to be added into this conference to make much more sense. So this is one photo to just express, okay, large animals, pigs are also treated in the laboratory this nicely. And finally, I would like to acknowledge these slides are not mine except last one. These slides are from the Senior Director Ella Monty Getiakami, and I would like to thank her. She has shared this slide for this kind of presentation or this kind of event. Thank you so much. Thank you.
So any questions, clarification? So I think with this we come to the panel discussion, and we have one more presentation. So I request Dr. Rajender Motiani sir, please come, and I also request from the audience Sweta and Garima to come and hand over the muffler and this our bouquet to Dr. Rajender Motiani. Everybody is busy with WhatsApp. Thank you, Dr. Rajender. Thank you very much. Now I ask both of you to release facility Dr. T.K. Atul sir. All of you, both of you come.
मैम साथ में आइएगा साथ में।
Now I request Dr. K.V. Patel, please come. Yes.
मैम साथ में इधर
So this is the last presentation, video talk by Dr. Kumar. The dogs could not come because he had some illness. So please bear with us. This is the last presentation, and then we will close.
Found in India and its neighboring countries, but they are found throughout the global south, and in fact in some countries which do not officially belong to the global south, for example in Russia and Italy. If you look at the evolutionary history of dogs, then dogs are said to have evolved from wolves or wolf-like ancestors into the dogs that we know today. And though we think of dogs as the pets which we have around us, actually dogs in their evolutionary course have existed not just as pets but as commensals around human settlements, and it is believed that dogs have undergone the domestication process through their interactions with humans, and the current theory is that dogs have undergone self-domestication rather than humans taking them on as pets. So the dogs that we see in our country, which are ubiquitously present around all human habitats, whether you go to the sea beach or to the cities or to the mountains, you will find stray dogs everywhere. And we feel that they represent how dogs lived around humans before we started breeding them and making them as our pets in the forms that we like to see them in. If we look at the humans and the dogs on the streets of India, we share a love-hate relationship. On the one hand, you will find people who actively feed dogs, or who give shelter to dogs, and of course, a lot of people who show love and care to dogs. But on the other hand, dogs also face a lot of threat from humans. We often imprison them, keep them in shelters, away from their homes, and of course, we cannot ignore the fact that dogs do bite humans at times, and India has a serious threat of rabies. When I look at my work in the perspective of the current world scenario, then we feel that our work fits into some of the sustainable goals of 2030. Goal 3, where we look at good health and well-being, because understanding dog behavior and ecology can help us to fight rabies. Sustainable cities and communities, because again, understanding an animal which is urban adapted, fantastically, we can think of building cities for the future which are friendly towards urban animals, and of course, life on land, because this is part of our terrestrial diversity. When we think of dogs' lives on streets, pet dogs, of course, receive all support and love and care from us. We help them breed, we rear them, we bottle feed them, we give them shelter, we give them vaccines. But then, when we look at dogs on the streets, they often live in large groups or small groups, and it is really the mother who is solely responsible for taking care of her pups. So, be it feeding them or keeping them clean or giving them shelter or protecting them from predators, it is primarily the job of the dog mother. During our work, we have tried to understand how much care a dog mother gives to her offspring. My first PhD student, Dr. Mohan G.H, looked into this, and to give you the story in very brief, we have seen that a mother shows different kinds of parental care behaviors, starting from suckling or nursing to cleaning them, which is allo grooming, sleeping with them in a pile to keep them warm, playing with them, protecting them, etc., etc. And if you look at the various colors here in these bar graphs, where each bar represents the data from one age class of the pup, so 3 weeks, 4 weeks, 5 weeks, and so on. And if we look at the proportion of time that the mother has spent in a certain kind of care, just by looking at the colors, you will see that the care levels or the levels of each kind of care changes as the pups grow older. And statistically too, we have seen that they receive extensive care and varied care from the mothers, and the mothers adjust the nature of care depending on the age of the pups. And indeed, as the pups grow older, if you look at the dots and the lines here, the red dots represent the active care given by the mother, which is energy intensive, like suckling and cleaning them up and play, whereas passive care is less energy intensive, but still is care because the mother is spending time with her pups, not going off foraging on her own or giving them protection from predators. So as the pups grow older, active care reduces, passive care increases steadily. Increasingly, as I said, dogs live in groups. Often, a group will have more than one litter born in one season, and we found that it's not just the mother who gives care, but often there will be other relatives in the group, female relatives, and sometimes males, which show care to their pups. Interestingly, the males which show care, we call them putative fathers because we don't know whether they are the biological fathers. And when we looked at the care shown by males in particular, we saw that males also distribute their time over the age of pups, showing different kinds of care, but majorly their care behaviors are play and guarding rather than other kinds of care or the varied kinds of care that the mother shows. Interestingly, it is very clear that there is a very strict window over which weaning happens. Weaning is the time when the mother gradually stops lactating and stops nursing her pups, and eventually they become independent of the mother in terms of their nutrition requirement. And our experiments and observations have shown that the 8 to 13 week of pup age is really the weaning period in the dogs. And we have also found that in spite of all the care that the pups receive, only about 90% pups born in a year survive, and the highest mortality really happens during the time of weaning, which is when the pups are becoming more and more independent, they're roaming on their own, they're going foraging on their own, and this is when maximal human-induced mortality happens. They undergo accidents, people chase them, beat them, they go into foreign territory, other dogs chase them, or people pick them up and drop them elsewhere, and these pups often get killed by the dogs in that territory, so on and so forth. And of course, there are people who poison and kill pups, not accidentally, but because they want to reduce the dog population. Now, that is, in very brief, the life stages of a growing dog. Now, dogs are scavengers primarily, which means that they are dependent not on hunting as their wild ancestors, but rather on leftovers from human sources. We carried out an experiment to first identify who the scavengers are in the typical urban ecosystem. So this was a very simple experiment where they dump a bowl of rice mixed with some gravy from either chicken or meat or fish dish cooked at home, or some dal, and we dump it and we record whoever visits the food source over the next 1 hour, and we were surprised to see that while of course there are dogs and cats and even goats and in one occasion a cow, the majority of scavengers were rodent birds. And you see here the pictorial representations of the different kinds of species that we got as our food source. When we did this over a large number of sites across the country, we finally looked at the data and we tried to identify who were these individuals of the different species who were the first to respond to this food source. And as you see here in this graph, the first responders varied from bulbuls, cats, the common myna dog, the house crow, the jungle myna, and the sparrow. These were majorly the scavengers, and they were always one of these species were the first to respond when the food was dumped. When the dogs were present in the scavenging guild at the site, then we found that they reduced the species diversity within a session, primarily because the dogs eat much more. An individual dog will eat much more in a given time as compared to, say, a common myna or a jungle crow. And so, when the dog came first, it pretty much finished off most of the food, and we did not get a very high diversity within the session, as you see here in this image. Whereas when the birds came first, then the diversity in the session was much higher. We did a lot of complicated analysis to identify the key species scavenging network. So this is a very rigorous network analysis using the species as a node in the network and the data from the different sessions that we ran on each one of each. And when we found, when we looked at the species, here is where the dogs lie. So each of these bands would be one species, and the dogs are emerged as key species of the scavenging network. What does that mean? It means that if you remove the dog from the network, then the connectivity of the network is reduced drastically. So this is just a representative image. We have done all that analysis by virtual exclusion, and the dog emerged as a key species. What does this mean? This means that the dog is definitely a very important member of the scavenging guild in the urban ecosystem. Now, the question arises that dogs, historically or biologically, they are canine, so they will be preferring meat as a food source. But we know that people feed them a lot of food-rich carbohydrates in India, that feed them rice and chapati biscuits. So we did an experiment early on when Master student Rajender did this experiment, where we were looking at food preference by giving them small pieces of bread soaked in different concentrations of chicken broth or water. And we found that the dogs are very smart. They will sniff first and then pick the piece which is soaked in the most concentrated chicken broth. And we say that they use sniff and snack strategy or SNS, and they will be first eating the protein-rich food and only then going for the less rich source of food. But this we found only in adults, not in children. And we thought that the dogs might be using a very simple rule of thumb. If it smells like meat, then you eat it. Now, Pravin joined the lab as a PhD student, and he was not just satisfied with this experiment. He said, but the dogs are really scavenging in a very noisy environment. There's a lot of garbage, and there's cleanup there, and then there is competition. How do we know that in real life they're doing the same? So he did what we call the dustbin experiment. You have 3 baskets. Each basket has a lot of garbage in it. One has 10 pieces of raw pieces. One has 5 pieces of raw chicken, 5 pieces of bread, and the third has 10 pieces of bread. Of course, there's a carbohydrate-rich box, there's a protein-rich box, and there's a mixed box. And he gave this box to the dogs, and the dogs were given just 1 minute to choose and eat. After we did this experiment with 96 dogs in different localities in Bengal and actually in Jamshedpur, we found that even when you give them this condition, individual dogs actually ate from the protein box more than from the mixed box of the carbohydrate box. And they were actually still using the rule of thumb. If it smells like meat, eat it. Now, Pravin gave them an even more complicated task. He added competition to already chaotic conditions of scavenging from garbage from these boxes. And he compared his data from groups of dogs versus solitary dogs being put to the same test. And when you look at the probability of sampling, then you see that the M stands for mixed, C for carbohydrate, and P for protein. And we found that when there are dogs in groups, then they sample less often than when they are solitary. So if they go to the either mixed or protein box, they'll try and find the meat and eat it, whereas for the solitary dog, they will take the time to sample all and then choose from the best, which already says that when there is competition, the dogs are trying to get to the food faster. And they were also spending less time being vigilant. What does that mean? They are not really looking around to see if something is coming, but when they are in a group, they are both going to grab the food as soon as possible. When we looked into the data into a little more detail, and here the probability of even eating was looked into. So when the dogs are in groups, they reach the decision faster than when they are solitary, which again means that they are being a little less choosy than when they are solitary. And dogs and groups are spending more time in each handling step, which is what it means that when they are in a box, they are not eating whatever they get and moving on to the next box, but then they are spending enough time to look for the food which is available and get it. Whereas when we are looking at the carbohydrate box, then we found that carbohydrate handling time was low, both in case of solid as well as in dogs. Now, Rohan put this entire data into a model, which is very interestingly a model which is proposed for scavenging strategies, and he said that the data that we get means the strategy, which is the optimal foraging theory, which has been tested theoretically in many cases, but hardly has been ever tested in a field condition where there is a balance or trade-off between the time taken to travel to the source, time taken to, or time spent at the food source, or the time taken, or when the animal decides to leave and go to the next food source, versus the energy that the animal gains by utilizing. So that is the trade-off, whether you spend more time in search or you spend a little time in search, get whatever you eat, get whatever you find and leave early and then search again. Or whether once you have searched, you decide to stay and eat as much as possible to maximize your energy gain. And this model that is pretty well known in theory is very nicely supported by the data that we get from our experience. Now we come to and their understanding of interaction in the urban ecosystem. Of course, we know that pets are very suggestions. People say that their pets understand their moods, they understand when pets come in, when the dog wants to point, they play with us, etc. etc. But then, all of this probably is possible because pets and they have learned through their time period of development to interact. Is it true of the stray dogs? The first set of experiments that we have done is in dogs with good understanding simple communication. You have bowls, both are covered, one has a reward, the other is sham-based, so it is smelling of chicken but does not have chicken. And then the experimenter randomly points at the bowl, and he checks the dog will go to the pointed bowl. What we found is pups are very ready to follow pointing, whereas the juveniles and adults, the chance of following is not following. You will see that they are following this suggestion. Interestingly, when you look at the cubs and you do trials for a dog, then you see the follow pointing gets rewarded, and then the response to the chance of them following pointing again is higher, whereas the follow pointing gets no reward, then we have negative ratings. And this is seen only in the adults, not in the juveniles, not in the followers. Interestingly, when we use more complex kind of pointing, which pet dogs to follow without training. In one case, there's a entry pointing, and then there is no cue. In the other case, the pointing is present, but from a distance, the dynamic is not pointing. We found that the breeding are better at following both of these than the dynamic proximal cue, where they tested them in the first experiment. And we feel that this is because they are accustomed to throwing food or showing them something from a distance rather than engaging with the food by putting it in a bowl near their mouth. So they are probably more apprehensive to bend low and point something, whereas when they point from a distance, they are quite good at following the gesture. This was quite remarkable because we could not expect the female dogs to do this because there was no training. These were dogs randomly picked from the street where we find them in the experiment and so on. The dogs don't know us, we don't know the dogs. Another set of experiment we did when we showed them friendly gesture, a low impact threat by raising a hand, and a high impact threat by raising a stick, not really doing them any harm, and then offering them a piece of food. And we tried to see whether dogs respond differently to these different movement postures. So one is called the social cue phase, the other is called the mobility. And that's a long story short. When you show them the positive social gesture, dogs will approach you, and when you give them food, they'll approach you more. Whereas when you show them the low impact threat, which is this many birds, they don't come to you when you call them. But after that, if you give them food, they approach quite often. Whereas in a high impact threat, even food does not hear them. So they are quite good at understanding the friendly versus threatening posture, again, without any training whatsoever. And they can change their decision for the same individual based on the cue they receive, whether positive or negative, and then with and not. So they are able to adjust their behavior depending on the cues they are receiving from unfamiliar cues. An experiment where we try to test whether it is food or the show of affection that makes dogs go. And we found when we give them food, one piece of food in hand, one piece of food on the road, and let the dog choose, most of them are going to choose the food in hand, even when we give them petting. When we do this experiment over a long time, the dogs here are fed from the road, and the dogs here are fed from the hand, and we can use them even after 14 days, the dogs don't really make contact. Whereas dogs who were given social food, depending on the head, no food. Then, large number of dogs change after 1 day of petting, and they keep on using food from the hand, showing that they understand the show of affection very much. In long-term observation, we also did to understand who are the individuals that dogs are interacting with, their group members, other group members, and humans, and we found that most interactions actually happen in private. Street humans occupy the bottom position, social networks. In a series of experiments, we carried out observations and then a test of approachability. Stretch your hand to the dog and call out to them and see whether they'll approach you. That is a test of approachability in zones of high human blood space, crowded places like stations, intermediate human blood space, like neighborhoods, some shops around, and low human blood space. A very peaceful neighborhood with very few people moving around peacefully. Firstly, we detect that different regions that we are then we look at approachability. Dogs in high and intermediate human blood approachable, less wary of strangers than dogs in low human blood regions. And this experiments were done in 9 cities across India, 200 dogs in total across 65. And for the first time, we tried to do something where we looked at the one-on-one interactions between dogs and humans based on the flux that they experience from day-to-day life. In continuation, during this field in West Bengal, in Kolkata, and in smaller cities of Rajender, during Durga Puja, there are a lot of people thronging the streets 24 hours around the clock in a stretch of a week or so, and there's a lot of food. So we asked, what was it happening? Will they be lured by the higher source, or the put out by the huge crowding that they experience in their territory? Understand this. 1 month before Puja, they would come, visited sites where we knew big, important pujas happen and attract a lot of people. And then we surveyed dogs. He went back to the sites during the puja, and again he went back 1 month later. So here we have abundance and human luck. Black squares are for pre-event, white circles are pre-event and red triangles are post-event. So you see here, pre-event and post-event data clusters together, and the event data is completely essential, where there is high human flux, no human check. So flux increased, food also increased because during the event, dog violence decreased. It shows the dogs really avoiding the crowding rather than being attracted by the excess food. And during the data, there was a also looked at the level of vigilant behavior and other behaviors that the dogs were showing during the census, and we found that during the event, the dogs are really varying. They are showing a high level of vigilant behavior as compared to pre-event and post-event. Pre-event and post-event data was 500. Overcrowding is a source of stress for the dog, which is, and the stress is so high that even the lure of food will not attract them to the crowded source. So you see that we humans, by our regular day-to-day behavior, indeed affect the lives of dogs in ways without even realizing it. So, if I sum up all that happened, breeding dogs have complex social lives, and this is what I'm trying to highlight in the first part. I was talking about the early life of the pup, because often we dog lovers would try to take pups away from their mothers, thinking that they are humans. Sometimes we have to take them out of our breed, because pups learn a lot from their mothers, they play a lot with their siblings, have very complex social systems, and during their development, a pup could be maturing into a dog, imbibing everything that it is experiencing through its interactions with dogs. They have to remember they are a key species in the scavenging guild in urban habitat, so moving down from the scavenging guild is going to some way or other impact the ecosystem life. They have complex relationships with humans and are highly dependent on humans. Dogs and humans have coexisted for centuries, and if we think about our own culture, our own literature, the earliest dog that I can think of is the dog in Mahabharata, which followed Mohan up the Himalayas and went to have education. If you think about this behavior, keeping everything out, then this behavior of a dog following a favorite human is something that every dog lover in India would have experienced at some point of time in their lives. And we have to remember that we have increased their evolution. So we need better policies to enable their coexistence in our habitat. I'm talking about eradication, putting them in pens, or adopting all of them, removing them from the streets, perhaps not the best solution that we can think of. I would like to thank
One more presentation is there. Okay, no presentation. So you see, Dream Team doing a very good work. So we have our Lagan team who is working with us, and after time, so I request Mateen, please come and offer the muffler and bouquet to our Ayana Krishnan. Very active person, Amit Misri. Come, sir. Now our my most favorite person, Bilal Ur Rahman, very great person. After contributing to our line, I request Dr. Kambos sir to please hand over the muffler and bouquet to Bilal. Very Priyanka. I request P. Atul sir, please hand over to Priyanka Bhairava. Priyanka.
She recently joined our lab from Guru Jambheshwar University, Hisar, Haryana. Yes, Sarkar. Anupama, can you come? To Dheeraj Sarkar, all time active, though he is fat, but he is very active. All your mail, Google form, all reactions, everything is done by him. And certificate also will be distributed by him. Thank you, Dheeraj Sarkar. We have Hari Shankar and Devangana and company, both of you come. Your friend also come. We have Hari Shankar and Hari. I don't know where they are. And she is very intelligent in animal related work. She got 10 out of 10 in her training program. And also your friend to hand over the bouquet. Hari Shankar idhar kaam kar raha tha? Bouquet and muffler. One person, Harsu, have left. So now, Aina will take.
So it's time to conclude the program with the national anthem. And I request our chief guest and all of you to kindly receive a certificate from the desk. So please rise for the national anthem.
मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलद तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जन गण मंगल नायक जय हे भारत भाग्य विधाता जय हे जय हे जय 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 हे भारत माता की जय भारत माता की जय भारत माता की जय we assemble for group photography. Smile please. No serious face. Behavior means always happy. आराम से फोटोग्राफर पूरा हो गया ना?